Good morning. My name is Anton Krucke. I'm the president and CEO of Tissue Genesis. We're a small company headquartered in Honolulu, Hawaii. And I think, although we're small, I think that what I'd like to show you is the amount of things that we've been able to accomplish as we're going forward. Let's see. Okay, take care of that business. So our work is in adipose tissue. So we're dealing with the fat tissue, and what we're going to do is we're going to extract out of the fat tissue what we call the stromal vascular fraction, which has the stem cell markers in it, the progenitor endothelial cells. So the fat cells are those round globulars. What we're really looking to sort out are all those things in between. And the stromal vascular fraction is proven to have a very rich source of regenerative capabilities, much stronger than just isolating one cell type. So the stromal vascular fraction, like I said, it has the mesenchymal stem cells in it, also the precursor endothelial cells and other regenerative cells. Our research and development and commercialization really started with the N45 patent out of Thomas Jefferson and the research by our chief science officer, Dr. Stuart Williams, and then we built a patent portfolio of over 45 different patents, not only around the cell mix, but the technology and the tools that we use to drive those and other methods. Okay, so everyone, in this strain has been looking at regenerative medicine. I think the companies that you're looking at are all taking different ways at this, and I think all of those activities kind of raise the water level in the potential for regenerative medicine in the field. We take all of our stem cells and other regenerative cells from the fat tissue. We use them immediately, so it's a non-cultured type of autologous application, take the immune system out of play. So we believe that we can produce a superior product, that our pellet will have a greater cell population, will also have a greater viability than what we see out in the marketplace. And how do we do that? We think that our digestion material is superior, and we understand we have a lot of knowledge about that, about 20 years of research and development. So we look at that digestion material before it comes in and before it's shipped out to the people using it. We have this, like I said, a high number of cells that we yield, over 10 million cells for 60 cc's, and then the whole system that we use is a closed system. Once you put in the fat, you push a button, you walk away, come back an hour and 4 minutes later, and you have the solution ready to use. Easy to use for operators, so it's built to be in the operating room. It's a very small system, sits on top of a desktop. This is what it looks like. It's really the only self-contained unit in its like for the adipose world. No breaks, no decisions, greater sterility. Okay, here's how it works. There's other things that work similar like this. We use a lipovage system as part of a filtering system when we do the liposuction. Once the liposuction is done, you take the fat over. The machine's been loaded by the operator, and once the machine's been loaded by the operator, you're able to put the fat in. We use a combination of the adipose tissue and temperature and also centrifuge technologies to isolate the pellet, right? And once that pellet is isolated, then the pellet is sucked out of the machine into a syringe, and then while the doctor's getting ready the patient ready, he now has the syringe ready to use for multiple applications that they would be applying to. So it's great to sit up here with a claim that we produce a great product, that we think that we know this business well. I think the way to do this is to go into accomplishments that do it, right? And so I'd like to talk about some of the things we've been able to do. We've had about 10 years of Department of Defense research contracts, and I think that the validation again and again that we can produce good data for them, can develop the technologies, has continued to be able to allow us to use this type of work. In addition to that, in the last 3 years, they recognize our capabilities in translating from a preclinical world into a clinical environment and have started to issue us contracts for work in a clinical environment. And they nominated our technology as one of the inventions of the year in the Department of Defense. In addition to that, this past year, in 2011, Tissue Genesis technology and our cell mix was chosen by NASA for the last space shuttle mission. So they've been studying in space for some time, and we've been working with them in command modules that you send up in space. And in this last space shuttle, they wanted to experiment and understand the effects of regenerative cells in a microgravity environment. And so we took our machine down there, we isolated cells, and then we put them in the command module and sent sent them up in space. Clearly, it's a fun project. We're going to do ongoing projects in the space station. But again, validation at a world-class level of the work that we can do and their confidence in our being able not only to execute an experiment in space, return it, and then yield the results. Other validation is we are the first company to be approved for an FDA clinical trial using adipose tissue with an automated system at point of care. So we've broken the ground for the other companies that are following us in this space, and I think that our experiences there and our work with the FDA will help all of us as we continue to go back and forth in trying to understand the platform that the FDA wants us to deal in. And so this particular trial that we were chosen for is a vascular graft. And so people with advanced PVD, where they would be harvesting vessel from one part of their body and put it in another part of their body, we would intervene in that process, and we use a scaffolding material, and then what we do is we coat the inside of the vessel with the patient's own cells. And then there's patented techniques around that coating method that we use. The picture there is a 3-month in a dog application that was part of the analysis that we did that got into the FDA trials. And we've been doing patients since March and working along there. It really, in addition to that, it establishes our footprint in the FDA with the device master file, and we have other applications that are in process with the FDA that piece, the device master file, has made that go very smoothly. And so I expect much more work in this area. I would expect by the end of this year we would be in at least we'd have 4 approved FDA clinical trials either by ourselves or by our business partners. That's pretty groundbreaking advancement. One of our business partners is American Medical Systems. They're now a wholly owned subsidiary of Endo Pharmaceuticals, and we've licensed our technology for them in the pelvic area. They have a lot of exciting things going on in their cell therapy backlog, and I would look for some announcements in the first half of the year on some very exciting things that we'll be doing together with them. We have been approved in Korea by the Korean FDA for the use of our technology in plastic surgery, and in 2011 we began shipping units and shipping our disposables. So with each unit you have the unit that's placed there, then you have a set of disposables and the enzyme solution. So we shipped 5 machines in 2011, 200 disposables, and we're just at the beginning of that commercialization there, and we're advancing the levels of approval that we have in Korea. So we have taken our technology on a world-class stage with the DoD, with NASA. We have approvals from the FDA. We have a pipeline now of applications into the FDA, and we've been approved for commercial work in Korea as we move forward. So that's our story. I really try to focus on, in this presentation for you, progress that's really being made. I think that when you can make these milestones or validation not only of tissue genesis technology, but of technology in the regenerative field, I think it moves the whole field forward. So with that, I really thank you for your attention, and I'm glad to be here. Thank you.